Good afternoon and welcome back to the Tech Talk series. CGI is proud to be a sponsor this afternoon. We are a global $10 billion organization with approximately 70,000 resources around the world, focused on high-end business and IT consulting, systems integration, application development and maintenance, infrastructure management, as well as having 150 IP-based services and solutions. Our commercial clients span the sectors of banking and financial services, retail and manufacturing, healthcare, and life sciences. In the U.S., our government clients include nearly every cabinet-level federal agency, all 4 military services, and the majority of the U.S. states. We've been part of the Pittsburgh market for over 30 years, providing services to some of the largest financial, healthcare, and manufacturing organizations in the region. We're proud to be a part of the local community and have further enhanced our presence with the merger of Summa Technologies. CGI's merger with Summa strengthens our capabilities for clients here in the region. With almost 400 resources in the area, we invite you to visit us at cgi.com. It's my pleasure to introduce you to our final speaker, Dr. Mark Collins, Vice President of Innovation and Strategy for Helomics.
Okay, so thank you very much for the invitation. As sponsor said, my name is Mark Collins. Robert Montgomery, unfortunately, is not here today, but he did all the hard stuff. So I'm just here to report on the work of my colleagues here. So when I was putting together the presentation, I thought that maybe I was being a little harsh in saying hype or hope about precision cancer, AI, and big data. So I thought I'd be a bit more positive and talk about the hope and the reality of precision cancer care as it relates to AI and big data. So what is precision oncology? What is precision cancer care? This is the hope, right? The hope is that we can take a bunch of patients with cancer, we can molecular profile them so we can sequence their genes, we can look at different things about their tumor and about them, and then we can use that information together with sophisticated analytics to develop prognostic markers, so markers that will tell us if you have this marker, how well you would do on a particular drug. Markers of drug sensitivity and resistance. Unfortunately, a lot of patients develop resistance to anti-cancer agents. And also markers of predictive events. If you have this marker, we shouldn't give you this drug because it will cause you harm. And obviously, most of the things about designing new drugs is that they don't do any harm to you. And then the idea is that we can take all those markers and use that to give you, the patient, the right therapy for your particular kind of cancer. So how do we go about doing that? This is the how, right? So the why was the slide beforehand. So the how really is, and this is an idealized case. This is how we'd like to see it done. It isn't always, as you'll see a little bit later on, the reality is somewhat a little bit different to that. But the idea is we take your big data, we take your genomic sequence, the sequence of your DNA and the sequence of the DNA of your tumor, most importantly. We take your medical record information, what drugs you've had in the past, what diseases you've had in the past, whether you had childhood immunizations and so on. We look at what medication you have taken and are taking currently. We look at things like the exercise you take, the environment, the exposome, as people call it, what you're exposed to during your lifetime, and also your nutritional status as well, what kind of stuff you eat. And then we take that, your big data, and we bring it together with their big data, everybody else's big data. And that data comes from, there's a lot of data out there regarding cancer profiles and so on that's developed from public data sources like the Cancer Genome Atlas, GEO. The All of Us is a research program funded by the NIH that's trying to recruit 1 million volunteers, US volunteers, to have their gene sequenced and their disease history followed for many, many years. In the UK, where I'm from, we've just completed one of those called the UK Biobank. Extremely large amounts of data, sophisticated data that we can use together with machine learning, statistical models, whatever kind of analytics, to tease out the data that shows us which biomarkers we should, which therapies we should give you for your particular kind of cancer. So that's the how. And there's no doubt that it works. There's just a couple of success stories here. So this is Roche has a new drug for HER2 breast cancer. If you have a certain biomarker profile, a certain test that you take, that drug will work for you in most cases. Keytruda is probably one of the most, you may have seen the adverts around. It's for non-small cell lung cancer, and it's one of these new immuno-oncology drugs where we're harnessing the power of the patient's own immune system to fight the cancer. And we use, whether you get Keytruda or you don't get Keytruda is dependent on a particular genomic profile that your tumor has. And then what's really interesting is now we're starting to see therapy based on not where your cancer is, but whether your cancer has a particular profile or not. And that's FDA approving Keytruda for any cancer, be it colon, lung, or breast, that has a particular kind of profile. So it's working. The reality, though, is it's working in a very small number of high-profile cases, right? So that's kind of the reality. That's a sobering thought that I want to explain a little bit more of, and how at Helomics we're taking a slightly different approach to precision cancer care. So part of the reality is that everyone's tumor is different, right? So back in 15 or so years ago, we thought that lung cancer was just one disease. Turns out that we can identify a bunch of mutations, variations in particular lung cancer, and there's also a lot of unknowns. So there's a lot of lung cancer tumors out there, but we don't know anything about them, right? So the reality of precision oncology is, while we can find variants, we can find profiles, right? We don't have the means yet to target the appropriate therapy. So you might get God forbid you get cancer, you go along, your physician sequences your tumor perhaps, may not be the case, and it comes back with a mutation, you have a mutation in KRAS or whatever. I'm sorry, we don't have a therapy for you, right? So there's a lot of way to go before we can actually marry all this information together and use it for the majority of cases rather than the exceptions out there. That's not to say that we're not making progress. I don't want to leave you with the impression that we're not making progress. We certainly are. So if you look at the numbers behind this is OncoKB. It's a database, a knowledge base maintained by Sloan Kettering. And you can see here that we have 476 genes that are variants in cancer across different tumor types and different drugs. But the idea behind this is, as a physician or a researcher, you can type in a gene and a variant and see if there's any approved drugs or otherwise that you could give that patient for their particular kind of cancer. And you can see here that the level 1, which is the FDA-approved drugs, the drugs that as a physician I'm allowed to give, is 14 genes. So if you hit any of those 14 genes, there would be a drug. All the way down to 28 genes where there's just biological evidence. We might have a result from an animal study or something. So we have a long way to go. Big data and AI will get us there, but this is a very, very complex problem. Who'd have thought cancer was complex? Right. So the reality of precision cancer care right now today is that it's sobering, right? It's still very good. It's a lot better than it was 20, 30 years ago, but it's about 30% of patients who get a targeted drug based on their particular biomarker profile, only 30% of those patients respond. The other 70% either don't respond, they have a toxic event, or they develop resistance to the drug. We're only looking at 5.7 months of progression-free survival. That's good, though. All right? And then if we look at the number of patients that where we can take their sequence, their profile, and target it to a drug, a couple of studies here, one's from MD Anderson, 6.4% of patients were able to be targeted to a drug, or 2%, which is this NCI match trial, which is matching patients not only with approved drugs, but with drugs that are awaiting approval, so in clinical trials. So at Helomics, we recognize that precision oncology and precision cancer care is a complex problem, and we take a slightly different approach, which I hope you'll see combines the best of big data and basic biology as well. So our mission is to be a leader in personalized medicine or precision medicine, whichever flavor you want to take, and to improve the standard of care for patients both now with the clinical diagnostic tests that we have, as well as in the future, using the data that we acquire from these clinical diagnostic tests to help research and target new drugs. So again, we've said cancer is, one size does not fit all. So what about if you could test the drugs that you would get, not on you as a patient, not use a patient as a guinea pig, because that's the reality, right? You get diagnosed with cancer, your oncologist says, well, the protocol for you is surgery, radiotherapy, chemotherapy. If you relapse, we'll do additional chemotherapy, whatever. So, but there's a whole menu of things that physician could give you and could do for you. So what we do at Helomics is we actually test the patient's tumor using the drugs that are approved to determine whether that tumor responds. So we take the patient's tumor from a biopsy or from surgery for removing a tumor. We grow those cancer cells up in the lab in Pittsburgh. We're right there in the chocolate factory with all the self-driving cars. We test them with a panel of approved drugs, the combinations and doses that would normally be provided to that patient for that particular kind of cancer. On the other side of the world, we take their tumor tissue and we deeply analyze it. So we run standard tests like histopathology. We run genomic analysis, so we sequence the tumor, looking for targeted mutations. We also do some biochemical tests and stuff like that. And then we evaluate all that data and we send the physician a report that details the results. And then that physician treats the patient based on the recommendations that we provide about which drugs work on the patient's tumor outside the patient's body, okay? Together with all the other targeted biomarker big data, if you will. So the reality of this, and these are clinical utility studies, you can go to our website and follow up if you want to prove that. So we can get a 50% improvement in progression-free survival by testing drugs on the patient's tumor outside the patient's body. We can get a 14-month improvement in overall survival. We get 2.5 times longer lifespan for the patient, treated with an agent that was shown to be active in the lab, as opposed to just randomly, well, not randomly, but just choosing the standard of care protocol. And again, because we're in a healthcare environment that's where costs keep growing, we can save money as well. And we specialize in gynecological cancers. So these are some of the cancers that are the hardest to treat. Ovarian cancer is particularly difficult to treat. Again, we can improve survival rates and so on using our technology. So what does that have to do with big data? We have a lot of high quality data. Part of some of the concerns about big data in precision medicine is that you've got lots and lots of different studies, you've got lots and lots of different organizations doing that work. They're not all sharing the data, they're not all using the same ways to represent the data, to tag the data, to annotate it. We have, from the very beginning, collected our data in a very specified way. So we have data about 149,000 tumors mostly, like I said, gynecological cancers, across multiple tumor types, multiple subtypes of tumors. We regularly measure multiple biomarkers, so multiple biochemical profiles of the tumor. Like I mentioned in the second or third slide about what kind of data we need to do this kind of work. We have de-identified patient data, including, in some cases, outcome data, so how well the patient did based on what we recommended. We have histopathology, which is like the gold standard. We bank all the tissue that we get from the patient in our biobank, so we can go back to it and analyze it in lots of different ways as new techniques come out. And we have multi-omic data, so that's genomic data, data about your genes, proteomic data, data about your proteome, all the proteins in your body, and so on. And what we're doing with that, and part of my job, Jeff, so my neighbor now knows what I do, is we're taking all this information and using machine learning techniques to generate a knowledge base of insights about tumor biology that we can use both to help inform our diagnostic platform, so we can feed that information back to the clinician to improve the diagnostic power of our tests, but also that we can provide that data to pharmaceutical companies for them to understand the relationship between different kinds of drugs, different kinds of biomarkers, different kinds of cancer, so they can generate the next generation of therapeutics based on a very rich set of big data coupled to, like I said, a machine learning engine that learns about the patterns in the data. And just to channel my inner geek, this is the basics of how DCHIP works. We have a bunch of clinical, demographic, and molecular data, both from the patient and from the public domain that we curate ourselves. We ingest that and de-identify that into a data lake. We have Elasticsearch to give us basic sort of searches and visualizations that we use. And then the machine learning engine learns the associations and the patterns in the data to provide insights out to a web front end that we put in front of our pharmaceutical customers. So this is not the dashboard. These are not the visualizations that we would give to an oncologist. We wouldn't do that. I think something like the talk from the Alexa guys would be kind of cool. The physician asks for the results and they got the report. But this is basic visual analytics. We can tell a lot about the data, give a lot of insights to customers about what drugs work with what biomarkers and what are the outcomes. So in summary, doing well. There's a lot of hope for precision cancer therapy, right? There's no doubt there's a lot of hope, and there are some very good examples of it working. What we're trying to do, and I think what the whole community, healthcare community is trying to do, and research community, is make that much more, the reality much more of a rule rather than an exception, right? Our approach has been for the last 10 years, is really to provide precision oncology right now with our battery of tests that, like I said, test known drugs on the patient's tumor. We've been able to take that data from a lot of patients, together with public data, well-curated public data, and advanced machine learning, there's a new term, advanced machine learning to build our DCHIP platform, and that DCHIP platform, like I said, is really designed to help inform current therapy. So we will feed back, we just started to do this with some of our major cancer centers, is feedback information about how this patient is doing in relation to all the other patients we've ever gone through our diagnostic procedure. So it's a bit like patients like me, this is like cancers like me, cancers like mine responded this way, to help inform the oncologist now. And then as you can see, we have this wealth of data that we can use to generate insights. We can dice and slice it in lots of different and interesting ways, together with our machine learning platform, to give insights back to pharmaceutical researchers to develop new drugs, do in silico types of experiments where they can test a particular biomarker to see if it works in silico using our system to design new therapies. So with that, I think I have about a minute left to say thank you very much. And if you're interested in learning more about Helomics, we are a Pittsburgh-based company, and visit our website, and I'll take any questions. Thank you. Sure. Yeah, so that was the slide that I listed. Yeah. So the numbers are, there we go. Get that one more. So we've treated about 149,000 people have gone through our chemo-FX test. So 50% improvement in progression-free survival. 14 months improvement in overall survival, right? And 2.5, yeah. Yeah. So I think the study was, the clinical utility study was 3 years.
So that's saying that 50% without that would have died in 3 years?
50% would have progressed to a more serious stage of cancer. So when we talk about progression-free survival, we talk about they survive and their cancer didn't get any worse.
Right. That's compared to the group that if they didn't take that specific drug would have been dead.
Would have been dead, or their cancer would have been worse. Right. Yeah.
I'm just curious, how long does it take for you to test the drug on the tumor outside of the patient?
It takes about 14 days. That's our turnaround time, which is about the turnaround time that you would get for most of the histopathology that determines which level of cancer you actually have based on sectioning your tumor. So we can turn it around in about 14 days. And given that that's a reasonable amount of time to get it to predict the best approach, rather than yes, you could. In other words, it's 14 days sounds like a long time if you've just been diagnosed with cancer, but 14 days giving you the wrong drug at day 0 and following through that course and it not being a good outcome, that's wasted time. So, yes.
Can your product be customized so that it can be used to treat other diseases, like other life-threatening diseases like cancer? And if yes, have you implemented it?
So the test that we have works on all solid tumors. So any solid tumor, not hematologic cancer like blood cancers. The machine learning that we use to do all the analysis of the data could be applied to other diseases like diabetes, or any time we have data and molecular profiles, we can in principle apply that approach. But it's specifically designed for cancer right now. Yes, please.
So would access to more data on more patients with cancer and treatment outcomes make this work better?
Oh yeah, and we, I say that kind of excitedly because we've been trying to work with the large hospital systems in our own backyard to get our hands on that kind of data because.
Are people collecting that data and it's just not available to you? Like, this is a logistical problem?
It's not a logistical problem, it's a scale problem, right? We can only do so many tests. Tests that are only paid for by certain insurers, and so on, that whole reimbursement thing that I don't pretend to understand. But by getting data on patients that haven't gone through our test, but have certain kind of cancers, certain kind of profiles, that's certainly valuable data to get, particularly if you can get outcome data as well. So you can, with machine learning, we can fill in the gaps where we don't have all the information. The more information that we have, the better, effectively. So the answer is yes. More is always better, as long as it's well curated and well understood and stuff like that.
So one of your slide has an unknown part in your So do you have any projected, number of tests you have to perform to complete this unknown area, or?
That unknown thing was that you see tumors with lung tumors with variations that you can't, how do I put it? You can't validate as being involved in anything. You see a difference in the profile between this patient's tumor and that patient's tumor, but you don't know what the driver is. So, and that's just because of the limitation of sequencing. It's a limitation of you don't have enough additional data to separate it statistically anyway. That's the problem with big data and genomics is, yeah, you'll always find a variation. It's whether you can act on that variation and it makes any sense. Apparently I'm keeping everyone from cocktails, so, so feel free to grab me at cocktails and I'll tell you more. Thank you very much.